My work on BRCA1 and BRCA2 started somewhat by accident. I'd been interested in the following problem, which is, why is it that so few early cancers in many epithelial organs actually progress to become invasive disease? And that led to the thinking that there must be mechanisms in our body whose purpose it is to prevent the progression of cancer. And perhaps if we studied families in which there was a gene mutation that accelerated the onset of cancer, we would get an insight into these mechanisms. I was lucky because at about the time my laboratory was being established and we started to think about this question, BRCA1 and BRCA2 were cloned as being the first tumor suppressor genes implicated in the genesis of these very common cancers like cancers of the breast, the ovary, and so on. And so obviously we started to work on BRCA2. And what we quickly found was that when you inactivate BRCA2, that it causes in cells which are dividing profound instability in the structure and number of chromosomes, which, as you know, are the packages in which our genetic material, DNA, is carried from cell to cell, from division to division. So having established that, we made the hypothesis that BRCA2 mutations and the loss of the wild-type second copy in the developing tumors might actually cause genome instability. This genome instability would increase the rate of mutations in these cells, and that would in turn lead to the development of early-onset cancers. And that really has been a major focus in our work over the last 20 years or so, which has been to try to understand how, when both copies of BRCA2 are lost, what happens to the genome, what are the mechanisms by which BRCA2 normally protects the genome from being unstable. And I think in the course of our studies, which of course have involved not just my own lab, but labs all across the world, we've now understood at near molecular resolution how BRCA2 works in the reactions that lead to the repair of DNA by an error-free process in order to preserve chromosome integrity. The second part of our work started somewhat later, and for a long time, most of us in the field believed that people who inherited one mutant copy of BRCA2 in their DNA would then lose the second copy in developing tumor cells in order for those cells to become cancers, causing genome instability, as I just mentioned. But we found to our surprise in an experimental model system just about 7 years ago that the second copy of BRCA2 need not be lost in order to get cancer. And this then prompted us to wonder, how is it that mutations affecting just a single copy of BRCA2 can cause cancer? Because that's not enough to cause genome instability. And what we found over the last few years is that certain types of BRCA2 mutations, the so-called missense mutations in which just one amino acid in the entire protein is changed, they work as what we call dominant negatives. So what these mutant versions of BRCA2 do is to inactivate or make it more difficult for the remaining normal copy of BRCA2 to function correctly, and so they lead to problems in cells which can, over the long term, lead to carcinogenesis. Just last year, we found, however, that truncating mutations, which actually truncate the very large BRCA2 mutations, work through a different mechanism. Interestingly, what we found is in cells bearing these mutations, DNA repair and other processes normally executed by BRCA2 work just fine. But when these cells are exposed to certain types of environmental and endogenous chemicals called aldehydes, the wild type level, the normal level of BRCA2 protein goes down. And because these mutant cells, cells from the carriers of these truncating mutations, already have lower levels of BRCA2, they become very sensitive to the effects of aldehydes, which in turn triggers genome instability. So we think that perhaps in carriers of truncating mutations in BRCA2, there may be environmental influences which initiate, promote, or otherwise modify carcinogenesis. I should emphasize, of course, that our findings are preliminary. They're based on laboratory studies. We have not verified them in model systems or, obviously, in patients. So they should be taken with a pinch of salt. But I think these observations do open the very interesting possibility that at least some forms of BRCA2 mutation may not predispose to cancer just by having deleterious effects in cells, but actually making the cells somewhat more sensitive to chemicals found in the environment and made within the body normally. And obviously this has important implications, we think, if it's correct, both for prevention, but also in finding new ways to treat patients carrying BRCA2 mutations. So aldehydes are everywhere. So for example, just looking around this room, aldehydes are found in the glue that holds furniture together. They're found outside in car exhaust fumes. They're found in cosmetics like shampoo. But I should emphasize that we don't think that exposure to tiny amounts of environmental aldehyde are likely to have a major risk in BRCA mutation carriers. The major source of aldehydes, which are very reactive small chemicals, is actually the metabolism of our own cells. And we wonder whether some of these metabolites, these aldehydes, may be made at different levels in different tissues. It prompts questions in our minds. For example, why is it that BRCA2 mutations confer susceptibility to breast, ovarian, pancreatic, and prostate cancer, and perhaps not cancers in other tissues? One possibility is these tissues may make more of these deleterious chemicals, the aldehydes, and that may help in part to account for tissue selectivity. Again, I emphasize that these are speculations yet to be verified, but I hope it gives you a sense of the implications of the work that we've been doing, trying to understand how it is that BRCA2 mutations lead to cancer susceptibility. That's really been the overarching focus of our work. I imagine a future in which we can prevent cancers from occurring in people with BRCA1, BRCA2, and other high-risk mutations that predispose them to cancer. I think there are two steps, one of which we have already made significant advances in, and that is treating cancers once they occur. Prevention, though, I think is and has to be the ultimate goal, not just for the sake of the patients, the carriers, and their families, but also in order to avoid unnecessary suffering and to ensure that people have an excellent quality of life, even if they carry these high-risk mutations. What will that take? Again, I think it's going to take a concerted team effort. From the perspective of basic science, as I've just been saying, my lab has really tried to focus on understanding how it is that carrying BRCA2 mutations predisposes to cancer. We still don't understand the very earliest steps in the genesis of BRCA2 mutant cancers. As I said before, people inherit one mutant allele. In some cells, the second copy is also lost. Why? Is it more frequent in certain tissues? What happens in cells from the time that they develop carrying a single mutant allele to the time that they lose that second allele? What factors, environmental, endogenous, genetic, modify this progression? As I also said, we now appreciate that a significant fraction of BRCA mutant cancers, BRCA2 mutant cancers in particular, do not lose that second normal copy. How do they develop? Do they follow the same set of cellular and molecular events to become cancers? Or is there an alternative course that they take? I believe that a thorough mechanistic understanding of these issues, the earliest steps in the genesis of BRCA mutant cancers, is going to be vital for developing totally transformative new approaches to prevention, which can then be taken through to translational and ultimately to preventive studies. For me, this is perhaps one of the most important areas of endeavor. And I hope that we will not only in the future be able to identify people who carry high-risk mutations, but intervene early to eliminate cells which are on their road to becoming cancerous, or even prevent them from striding down that road in the first place. I think the Basso Center has already had an enormous effect. First, at UPenn itself, it's clearly, even from my brief experience, created a family, a community of researchers, clinicians, patients, genetic counselors who share the vision of the families that founded the center. I think that's tremendous because it speaks to the issue that we're not going to tackle the patient's problems in any one silo, but rather through a coordinated effort that transcends any given specialty. It's obviously a very singular honor, but I received it with somewhat mixed feelings. On the one hand, there's obviously an intense sense of pride that we've been able to accomplish something which is recognized by our peers, of gratitude to the Basser, Gray, and Potter families for having endowed this prize and indeed endowed the Basso Center, but also of humility. This was not just my work, obviously. It was the work of many people in my laboratory over the years. And moreover, we built on the observations of many other scientists, researchers, clinicians working across the world. So I can kind of paraphrase my feeling on this last point by saying the following, which is, of all human activities, human creative activities, science is perhaps the one that is not really an individual act, but one that depends on the work of many people. And paraphrasing my much better known Cambridge colleague, Isaac Newton, from long ago, I might say, if I see further, it's only by standing on the shoulders of giants.